Alright, so in another relatively short recording, I want to address the concept of DNA packaging. So what we want to remember is that the state of the DNA in the cell can change. Now, in prokaryotes, generally we have a circular chromosome, and it can be supercoiled, and it tends to be kind of sequestered in the middle of the prokaryotic cell, though not membrane bound, in what's called the nucleoid region. It is associated with a small number of proteins, but not nearly the number of proteins that we see associated with DNA in eukaryotic cells. So we generally think of prokaryotic DNA as basically being naked as compared to truly packaged DNA that we see in the eukaryotic cell. So let's kind of walk through the packaging that we see and that enables us to get almost 2 meters of DNA into a human cell. So again, the amount of DNA that you have per cell is species specific, but for humans, 3.1 billion base pairs is going to stretch out to about 6 feet, right? 2 meters. How do you get that into a 50 micron cell? Well, it has to be packaged pretty specifically. Now, not only that, but we also are going to use the packaging of DNA as a mechanism to control access to the information in DNA. And we're going to come back and talk about this later when we talk about control of gene expression. But if I have something wrapped up and packaged away, I can't get the machinery at it to read the sequence. And I can use that then as a control mechanism to say these genes, which are packaged away, are not going to be read, they're not going to be turned on. Like I said, we'll come back to this later. But if we start now at the basic level, here's our double-stranded DNA. We're going to wrap that DNA in eukaryotes around proteins called histones. And the histone proteins with the DNA wrapped around it comprise what's called a nucleosome. Right? And so when you string all the nucleosomes together, you talk about this as like the 10 nanometer fiber of DNA. And you can literally see that in those electron micrographs, little kind of beads on a string. And that's DNA wrapped around protein. Now, you can take that 10 nanometer fiber and wrap it around itself to get a 30 nanometer fiber. And then you can take that 30 nanometer fiber and start to loop it out from protein scaffolding. And that protein scaffolding becomes ultimately a very, very thick 300 nanometer fiber, which then can be folded back and forth upon itself to make what we think of as the condensed chromosome that we'll see in metaphase of cell replication. All right. So all these levels, at this level, there is no DNA that is accessible for expression, but it is an incredibly efficient packaging mechanism. So we want to kind of think about that. Chromatin is organized into fibers: the 10 nanometer fiber, that's the nucleosomes, the 30 nanometer fiber, 300 nanometer fiber, and ultimately the metaphase chromosome. Now, what we think about when we think about chromatin, i.e. the general term for DNA in the nucleus, we think about it as sometimes called like kind of spaghetti in a bowl. And that's a metaphor that I'll use, or an analogy that I'll use oftentimes when I'm teaching it, and it works fairly well. But I do want you to understand that it's not entirely accurate. So there is more organization. There are some chromatin that is packed more loosely, and that's called euchromatin. And there are some regions of chromatin that are packed much more densely. It's called heterochromatin. And what it suggests when we consider these two forms of chromatin, or DNA in the nucleus associated with proteins, is that there is some organization to that quote-unquote spaghetti in a bowl. And so when you look at the different chromosomes as they're kind of in their diffuse state, in the chromatin form of the nucleus, we can see that there is distinct organization of those chromosomes, and we believe that this is going to contribute then to the control of gene expression from what we call distal control factors, and we'll talk about that more later. But the idea is that you can have something that's very, very far away from a gene, nucleotide-wise, very, very far down the chromosome, or potentially on another chromosome, that actually controls the expression of a gene that is very far away. So know that we think about DNA in the nucleus as kind of spaghetti in a bowl, but there is organization there, and if you understand that, that's good enough for now.